Good afternoon, everyone, and thanks so much for joining us today. I'm Leonie Stevenson-Jones, and I work as a grants officer for the Alzheimer Society, who, along with Alzheimer's Research UK, funds Brains for Dementia research. It's my pleasure to welcome you here today to our series of BDR engagement events. The purpose of these events is to share the ongoing work of BDR and also to thank you, because none of it would be possible without you, our study participants, partners, and your families. We did have plans to hold the events in each centre last year and this year, but unfortunately with the ongoing pandemic, we've had to switch to holding them online. We're recording this event and the others in the series so that you can watch them back or catch up if you're unable to make any of them. Today, you'll be hearing from Phil Tinkler, who coordinates the Manchester Brain Bank, Fred Walker, a BDR participant, and Dr. Gemma Lace, a senior lecturer at the University of Salford, who has used BDR tissue throughout her research. They will each give a talk on their involvement with BDR, and then there'll be a chance at the end of the session for you to ask your questions. You can find the place to submit your questions in the black bar at the bottom of your screen. Click on the Q&A button and another box will appear where you can type your questions. You can submit your questions at any point during the session, and when it comes to the question and answer time, I will pop back on the screen and be putting them to our speakers. So now I'd like to introduce Phil Tinkler, coordinator at Manchester Brain Bank, who will share some updates and insights from the centre. So over to you, Phil.
Thanks for that, Leonie. As you've just heard, I'm Phil Tinkler, the coordinator here at the Brain Bank at Manchester. And yeah, I'm just going to give you a quick overview of the Brain Bank here before we get on to Fred and Gemma, really the main talks of the event. The Manchester Brain Bank was established in 1980 by Professor David Mann. So it's been going for quite a while now. And it's headed now by Federico, and we're primarily funded by the Brains for Dementia research project. So it's a huge part of what we do here in Manchester. Next slide, please. So like the other brain banks, our main focus is on research. So we have our donors who have donated their tissue to us. My colleague Andy, he prepares the tissue for research, and we conduct our own research within the organisation itself. But a big part is also collaborating with other people outside of the university. So doctors and academics from all over the world can apply to use the tissue and data that we have. And we have a committee set up to review each application. So we look at the ethics, the science, the practicality, things like that. And those that tick all the boxes, we will release the tissue and data. So while we do our own research, a big focus is on collaboration and working with other people as well. Next slide, please. So you can see here just a bit of a breakdown of the different donations that we have at the Brain Bank here in Manchester. So you can see primarily we've got people with Alzheimer's disease, but lots of other types of brain disease as well. And importantly, people who are controls, because it's very important to have someone who is healthy to compare against someone, for example, with Alzheimer's disease, so we can see the differences between the two. So that just gives you a little idea of the breakdown of donations that we've got. So next slide, please. So in Manchester, as part of the Brains for Dementia research project, we've got 538 people who've signed up since 2009. So some people have been involved for a long time. We've had 159 people who've actually gone on to donate their brain to the Brain Bank so far. And then 261 people who are seen each year for the assessments. I know some of you have dialed in today will be participants who do the assessments, some maybe even with me. And yeah, that is the crucial part of Brains for Dementia. It's having that information, the assessments with people for over 10 years sometimes, things like lifestyle, medications, any comorbidities, memory, mood, all that information while someone was alive. And then when they pass away and donate their brain to us, we have the brain donation as well. And it's those combined two things that are invaluable for research, and that's what makes the Brains for Dementia project so important. So next slide, please. So yeah, it's been a difficult year for everyone, and sadly the brain bank didn't escape the effects of COVID. So we actually had to stop taking donations back in March 2020 at the start of the pandemic, but we've since reopened in September, and I've been working with families to make sure that the donation goes ahead. And since September, it's been going really well, and we've been getting people to be able to donate to us. The other part was the assessments that I mentioned before, so the yearly memory tests and other questions. We've had to do all of those over the phone due to the virus. So, you know, those of you who've dialed in and you do those assessments, thanks for bearing with us there. I know it's difficult to remember 10 items at the best of times, but trying to do it over a crackly telephone line is extra difficulty there. So thanks for bearing with us for that. And we're hoping to return to being able to offer face-to-face visits soon, once vaccinations and things like that have come into effect. And we're going to prioritise people, for example, who hearing meant that doing the assessments over the phone just wasn't really possible. So yeah, we hope to be able to move to doing that soon. So next slide, please. So lastly, yeah, I just wanted to say a big thank you to everyone involved, especially our participants on the Brains for Dementia research project. You know, thank you for sticking with us, and I know some of you have been involved for years and years now. So yeah, a big thank you to you all. We've got an excellent talk coming up next with Fred. So he's actually one of our lay committee members. So as I mentioned before, the people who review the applications to use the tissue and data we've got at the brain bank. And he's very experienced with dementia, involved with quite a few things. And he's going to go through his own experience of his wife's journey through Alzheimer's disease. So I'll hand over to Fred now, and thank you very much.
Right, I've got my notes up. Would you let me know when slide 1 is on?
It's up, Fred.
Right, thank you. My name's Fred Walker. I'm 83 years of age. I'm a retired engineer and a champion of Alzheimer's Research UK. This slide is a photo of my wife, Joan, who I met at the local dance hall about 60 years ago. And given it a coat of thinking about, I used to go every Wednesday. So 60 years may well be tomorrow evening when I met my wife. We actually courted and married. We had 3 children of our own, and then we became foster parents. And we fostered 30 newborn babies who were going for adoption. And we would get the babies at a couple of days old, keep them for 6 weeks, and hand them on to the adoptive parents. We were also regular churchgoers and joined the parochial church council, passed advanced driving tests, and were members of the Institute of Advanced Motorists. And Joan was the local secretary of the group. We enjoyed fell walking and all other outdoor activities. So we had full and active lives, both mentally and physically. Now we know that early on in Alzheimer's disease, the patient tends to lose navigational skills. And this happened with Joan. Sometimes I'd arrive home from work and there'd be a note on the side saying, "Gone for a drive, see you later." Except on this occasion, she went for a drive, but I didn't see her later. 10:30 at night, I get a phone call and she's in Weston-super-Mare, some 200 miles from where we lived. She'd obviously dropped onto the southbound M6 and kept going until she saw a road sign that she recognised. Having been brought up and bred in Cheshire, she knew every road sign all the way down, but she just did not recognise them. She then had a series of falls, and each time she cracked her head, so each time I would take her to A&E. After the third fall, A&E referred her to the Falls Clinic, where she was given a scan, and forwarded on to the Dementia Clinic, where she was diagnosed with Alzheimer's disease with Parkinsonian symptoms. She continued to have falls, but by now these falls were accompanied by fitting and vomiting. Other symptoms began to develop. Now we all know that when we put a stamp on an envelope, that stamp goes up at the top right-hand corner. But Joan's hand-eye coordination was gone and the stamp could appear anywhere on the envelope at all. Her favourite way of leading into Christmas was to write the Christmas cards, but she then said that my handwriting was better than hers and I should write the cards. It turns out that she was forgetting how to write and she couldn't even make a signature on a cheque. She developed aphasia, so instead of saying something like, "Fred, shall we have a cup of tea?" she'd say, "Fred, shall we da 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 da?" And all that would come out is a progression of plosive sounds, so I had to finish the sentence for her. If she wanted to go from the lounge to the kitchen, she would go to the lounge door. If it was shut, she would just stand and look at it. She'd be unable to open the door. So I'd open the door, but then she was unable to go through because the colour and texture on the other side was different and her brain couldn't process the information. So as an engineer, there's always a plan B. And on this occasion, the plan B was to sit her in a wheelchair, get eye contact, talk to her, and propel her through the doorway backwards. Once we were in the new area, that then became the norm, but she wouldn't be able to return to the lounge. Slide number 2, please. Now, when you're out walking your dog, playing golf, or just walking in the countryside or jogging, if you come across a rabbit hole, would you stick your hand down it? And the answer is probably no, because you don't know what's down there. But someone with Alzheimer's, there's no difference between that rabbit hole and a sleeve hole. And I had terrible job getting Joan to dress because she just would not put her hand into the sleeve of a garment. So my plan B at the time was to dress her from the back. I could guide her hands into the sleeves, lift the garment up onto her shoulders and fasten down the front. Back to slide 1, please. We then had problems with bathing because we found that she couldn't balance on one leg and then cock her other leg over the rim of the bath to get in. And she was frightened in case she did get in, she'd never get out. So we changed the bath for a shower. Now the shower is a gravity shower. It's not one of these power with needle sharp spray. So the water just laid on very, very gently, and she was okay with that for a while. And then she began to get more and more agitated. So next time I had a shower, I analysed everything that was going on, what I could see, hear, taste, smell, feel, everything. And the only thing I could come up with was that when water runs off over dry skin, it irritates. Over wet skin, it doesn't. So again, an engineering plan B. Instead of showering Joan from the head down, I would start at her feet and go upwards. That way, all the water was running off over wet skin and there was no problem. Now, the change from continence to incontinence can take some considerable time. So it meant that trips to the toilet became more and more frequent. On a good night, if Joan wanted the toilet 5 or 6 times, that was great. On a bad night, I'd get her toileted, back into bed, and by the time I got round to my side of the bed, she was saying, "Fred, I need the toilet," and we'd have to start all over again. On one occasion, she asked for the toilet, and I went round to her side of the bed, but in the meantime, she'd gone into a full body spasm. So she was stiff as a plank from head to toe. So the only way I could get her out of the bed was to turn her across the bed through 90 degrees. Now, I'm sure when you do that, your knees bend and your feet flop down to the floor. But with a full body spasm, her feet were sticking out into free air. So the only way I could get her out of bed was a dead weight lift, a hoist out of bed, which I did, and I had to hold her very, very close, otherwise she would have fallen over. And it was then that the enormity of this terrible disease hit me, because instead of a robust woman, I've got this frail, vulnerable, utterly dependent person who was totally different from everything that had gone before. So I came to the decision that there wasn't a hospice or a hospital that could give her one-to-one, 24/7 care and attention that she needed. So I was determined to keep her with me until the very end. From then on, my role changed from carer to nurse, and as such, I discovered she had a blockage in the bowel. She went into hospital for diagnosis, where they diagnosed ovarian cancer, and the cancer was adhering to the valve and causing the blockage. They gave her between 4 and 6 months to live. While she was in hospital, I had a hospital bed delivered and set up in the lounge against the patio doors so that she could lie there and look out over the garden. Now, with Alzheimer's, the patient tends to forget, the memory goes, and they forget names, dates, faces, even a spouse of 50 years. But the executive memory goes as well. They forget how to use the phone, the cooker, how to walk, how to talk. And in Joan's case, she even forgot that to eat, she had to open her mouth. So I had to blend the food, load a spoon, and gently work it between her teeth to deliver that food. Unfortunately, she'd also forgotten how to chew and swallow. So I'd have to massage her throat and neck to try and get the muscles stimulated to swallow the food. Of course, that didn't work. At the end, all I could do for her was to give her morphine for the pain from the cancer and to moisten her lips to hopefully keep her mouth nice and fresh. On the day she died, we had friends in to visit, and I was chatting to them at one end of the lounge, and Joan, of course, was lying at the other end. And I don't know where the message came from, but I knew Joan needed me. So I asked our friends to leave. I took Joan in my arms, I stroked her hair, and I gently talked to her. And as I held her, I watched the pulse at the base of her neck, and I watched it slow and finally stop. I carried on talking to her for a while, and then laid her back because she'd come to the end of her journey. But it's a journey that no one should have to make, not as a patient, not as a carer. After she passed away, I had a decision to make. Do I walk away from this disease or do I fight back? Can you remove the slide, please? Now, there are many, many charities looking after patients and sufferers in the community, but there's only research that can really help them by finding reliable diagnostics, preventative drugs, and a cure. So I joined Alzheimer's Research UK. I put together a PowerPoint presentation and toured the clubs and societies like Roundtable, Women's Institute, Probus. I was even asked to give talks at Manchester, Salford, Edinburgh universities. I was asked to join BDR and the Manchester Brain Bank, and I donated my brain. Now, the brain donation process involves an annual assessment, as Phil has already mentioned, and that assessment is recorded and the volunteer is scored. That assessment can actually last upwards of 2 hours and covers all aspects of brain function, blood pressure, and perambulations. My next assessment is actually in June with Phil. And it's a bit like an MOT on a car. I think you'd rather go to the dentist than take the anxiety of a test. But that's where I'm up to. I'm really looking forward to a new assessment, not. Now, as an engineer, I view the body as a machine, and I'm interested in its progress. So there's two sides of my coin. And when I die, my brain will be harvested so that the researchers will have the brain, but they'll also have a record of the lifestyle that that brain could support. And that makes up a solid gold research tool. Now you may ask, why is a retired engineer invited onto the BDR and Brain Bank committees? As a lay member of BDR and the Brain Bank, I look at the ethics. So when I see an application, I treat it with the utmost confidentiality. I also check for equalities because age, gender or ethnicity, etc. must not influence the application. All applications for tissue, bloods or data can come to BDR and the Brain Bank from anywhere. We have applications from Singapore, Southampton, New York, and they carry both the technical and lay descriptions of the research program and its benefit for patients. There are occasions when a researcher can spend 20 years in the lab researching Alzheimer's disease, but never see a patient. I've had 20 years as a carer and nurse for Joan. I also mix with patients and their carers in my work for Alzheimer's Research UK, so I can bring that experience to the table. And finally, I asked myself if Joan would be happy for her tissue, blood or data to be used in this particular programme. And of course, I asked myself the same question. I can then adjudicate on the application. Over the years, I've found that Alzheimer's is different from most other diseases. With other diseases, you can hold a conversation with the patient. You can reminisce about the good old days, last night's telly or what you had for breakfast. But you can't do that with Alzheimer's. This disease destroys everything that makes that person a person. I've often heard it said that Alzheimer's disease is like a second childhood, but nothing could be further from the truth. A child is a growing, developing person. Someone with Alzheimer's is disintegrating. A child loves to have its nappy changed, but an adult with an incontinence pad is ashamed. It's so easy for them to lose their dignity, but they will only lose their dignity if we stop treating them with dignity. They may not remember what you said 5 minutes ago, but they will remember how you made them feel. They'll remember love and joy, kindness and consideration. And when they finally pass away, it's not a traumatic event. It's just another step in this terrible disease. They pass gently from one state to another. You grieve, of course you grieve, but you've been grieving for 6 years and grief has become a way of life. And what I also came to realize was that Joan was worth so much more than what she could remember. Well, that's the end of my session. I want to thank you all for listening, and I'll hand you over to Gemma.
Thank you so much for handing over to me. That, you know, every time I hear about your story, Fred, it just gives me a lump in my throat, and I always worry about having to speak when, you know, after you, because I think it's amazing. Okay, thank you. Just got a bar that's just blocking my view from being able to actually start the slideshow. There we go. Okay, so thank you so much to the organisers for inviting me to come and speak today. So my name's Gemma, and I'm a lecturer at the University of Salford, so I lead a dementia research group. And today I want to tell you a little bit about how Brains for Dementia Research and Alzheimer's Research UK and the Alzheimer's Society has actually made the research that my team does possible. And I'm going to be talking to you about brain junk today. So, you know, my first slide is what is dementia, and Fred has just really beautifully captured the personal side of dementia. And we know that dementia really, it's an umbrella term. It describes a set of symptoms, and these symptoms can be variable. You know, the ones that we often think about are memory loss and difficulties performing cognitive tasks, personality changes. And we know that what drives dementia, this set of symptoms, are diseases. And there are lots of diseases that are known to cause dementia, Alzheimer's being the most predominant form of dementia, also frontotemporal dementia. And whilst these different types of dementia manifest themselves differently clinically and affect people differently, actually they have some shared features as well, which when we come to study those diseases, it's useful to reflect on both the similarities and the differences. So an example of this is if we were to look at a sample of brain from a normal healthy individual and compare that to someone with a type of dementia, you can see that there's a marked difference. So on this slide here, you can see on the left, you've got a normal brain. On the right, you've got a brain from someone with frontotemporal dementia. And you can see the brain from someone with frontotemporal dementia is much smaller, it's shrunken. And that's because brain cells die in dementia. And this happens in Alzheimer's and a whole array of other disorders that cause dementia. And what goes alongside that brain cell death is a commonality, if you like. So whether we're thinking about Alzheimer's or frontotemporal dementia, you get the buildup of brain junk. So you get this abnormal buildup of proteins, and we know that these proteins are very damaging to the brain. They cause the brain cells to die. And in an adult, unlike other cells of our body, brain cells can't just replace themselves, and circuits, the complex circuits that these brain cells form that allow us to be who we are, get disrupted. And so if you look at the image on the left-hand side of the screen, at the top, you can see a section of brain tissue from someone who is healthy and aged. And the important thing to know is there's no brown stuff, right? And that brown stuff is the brain junk. Whereas if you look at the frontotemporal dementia and the Alzheimer's disease brain sections, you can see some brown stuff. And this is the abnormal protein. Now, some of this junk, some of these proteins are seen across a huge number of different types of dementia, whereas other forms of abnormal proteins are very specific to a certain type of dementia. What we do know is that in a healthy person, you know, these proteins don't build up as much. And so there's lots of evidence that these proteins are damaging, they cause the brain cells to die. And so researchers like me are very much interested in this process, because if we can stop that protein from building up and damaging cells, we can stop the brain cell death. And the more that happens, the more of the person we can preserve with respect to the dementia. So why doesn't junk build up in the brains of a healthy individual then? Well, just like at my house, I've got a black bin for general waste, and I've got a blue bin for paper, I think, and I've got 2 brown bins for plastic and glass, and a green bin for garden waste. Our brain has got a number of different bins, waste disposal systems, who have roles in degrading certain type of junk in our brains. So like we've got a black bin, or in this image, a green bin, which kind of, you can put any old junk in. Our brains have got the equivalent of a black bin, where it will degrade anything that's potentially damaging to brain cells. Whereas other waste disposal systems in our brain are very precise. They will only degrade proteins or cell components that have been marked for degradation. Others will only degrade a very specific subcomponent of a cell. So there are lots of these different waste disposal systems that have got different jobs, but ultimately they all work together to make sure that this junk doesn't build up in our brains and doesn't cause enough damage so that you end up with cell death. So what my research team are really interested in then is why is there this buildup of junk in the brains of people with dementia, and why does that happen? You know, if you think about waste disposal as a mechanism, it's quite a complex system. So first, rubbish has to get to a bin. It needs to be selected and identified as rubbish. It needs to get to a bin. It needs to be sorted so that it goes in the correct bin. And even if we are the most efficient recyclers and we are fabulous at putting our rubbish in the bin, well, if the bin man doesn't turn up to empty those bins and clear those systems, then you're going to get a spillover and you're going to have a knock-on consequence to that. So I don't know about you, but I had a failed bin delivery a few weeks ago, and it caused absolute carnage in our house, where there was waste everywhere for a while and we had to bag it up and we didn't know what to do with it. And our brains are the same. The systems can get overwhelmed if the normal sequence of events don't occur. So we're interested in why this junk builds up in the brains of people with dementia, and also importantly, what is going wrong. And the other thing that my group are interested in, so I'm a bit of a jack of all trades, master of none. I'm interested in all different types of dementia. I'm interested in Alzheimer's, frontotemporal dementia. Also, I'm interested in Parkinson's disease, and have worked in that field for a while in my younger days. But what I'm interested in is we know that some junk you see across a whole spectrum of dementias, but others is only very specific. So does brain junk accumulate in the same way for the same reasons in different types of dementia? So we know that there's a general problem in the degradation of brain junk, but why is that different in different types of dementia, and how is it different? And what does that tell us about the disease process? So why use human brain tissue then? Well, there are a number of different ways we can study diseases such as Alzheimer's and frontotemporal dementia. There are animal models of disease where you can make animals that have a mutation so that they resemble the human condition, but they don't really resemble the human condition. And we can do cell-based studies where we can grow brain cells in the lab, we can manipulate them and then and measure things at the other end. But the important thing, and this really links back to what Fred was saying, is that, you know, the individuality of dementia and how it affects people differently. You know, individuals with dementia have led individual lives where they've had individual food and drink preferences. There've been individual exposures to viruses, brain injuries, maybe exposure to toxins or medications or other pre-existing medical conditions. There may have been variations in the kind of lifestyle choices that were made, and maybe individuals had different stress levels. All these different variations are also on the back of difference of genetics, individual variation in genetics. And so, you know, this mishmash of environmental factors and genetic backgrounds is only captured when you look at an individual's brain. You can't grow that in a lab. And so if we are to really understand the uniqueness of what is happening in the brains of people with different types of dementia, we need to look at the full picture. We need to understand about how all these factors work together to end up with someone developing dementia. And importantly, and Fred alluded to this as well, we need not just to focus on the late disease and what goes wrong in the diseased brains, but also look at the healthy aging brains as well. What is it that makes those brains so strong and resilient to disease? What are the early stages of disease that trip someone and put someone over the line from having a healthy aged brain onto the pathway of developing dementia. Because if we can understand what switches are being activated in the early stages, then we can maybe do something to prevent someone from going down that process. Because we know that actually this buildup of junk in the brain happens decades before someone actually develops symptoms, but we do know that it puts them on the path to developing dementia. And that's why human brain tissue is such an unbelievably informative resource. It's the only way we can really get a full and rich understanding of the individual nature of dementia. And so this is what we use in our group. So we do supplement our work with cellular-based studies as well, but it's the human brain tissue that kind of generates this huge body of information. So how does it work then? Well, as a scientist, I have to apply for funding, and people like Fred will scrutinise, and experts in the field will look at the funding application put in for. I will decide what experiments I want to do and what hypothesis and ideas I want to test, and then I will send that off, and then I will either get a yay or a nay. And I was very lucky to have my work supported by Alzheimer's Research UK and the University of Salford, and also Brains for Dementia Research, which is what gave me access to brain tissue as a resource. Using that funding, I was able to recruit 2 absolutely amazing young students who were very, very passionate about progressing dementia research, and also very amazing advocates in raising dementia awareness. So we've got Richard here, whose work focused on frontotemporal dementia, and we've got Diana here, whose work focused on Alzheimer's. And what these guys did was they got these brain samples and they stained them so that we could look at them down the microscope. And the first kind of set of images in the center of the slide show the brain junk. So you may have heard of some of these brain junks. So tau is an abnormal a protein that you find in an abnormal form across a whole array of different disorders called dementia. And beta amyloid is another type of brain junk which you only really see in Alzheimer's. So we can look at the junk, and also we can stain the brain, and we can look at markers of these waste disposal systems. Now, these waste disposal systems are very, very complicated, and they've got different components, different sets of machinery, but we can look at those and we can use that information to try work out where things are going wrong. So I didn't want to make this this talk really heavy on the science, but I just wanted to pick out one of the key findings that has popped up throughout the maybe 5 years or so that this work has been going on. So what we did was we looked at brain samples from people with no disease right up to severe disease, and the red marker that you can see here, this is a marker of a really important component of our black bin, our general waste disposal system. And what you can see is the amount of redness, the amount of this marker decreases with increased disease severity. Whereas when you think about the junk, so in this case, the junk that we're interested in is this tau protein, as disease gets more severe, there's more of it. And so basically with advancing disease, you get the buildup of junk, but the markers of these waste disposal systems, who are supposed to be functioning to get rid of the junk, are going down, which is again supportive that you've got this problem. And the human brain tissue is what allows us to look at more than one thing at once in the background of all the various clinical information that has been provided as part of the brain donation program. So what have we found overall, just to kind of try box things up a little bit? Well, importantly, what we found is actually that in the different forms or different diseases that cause dementia, so when you compare Alzheimer's to the different types of frontotemporal dementia, which there are a few different types, the deficit is different. So the problem with respect to waste disposal is different. You know, in Alzheimer's, it seems to largely be the problem of the black bin, whereas in frontotemporal dementia, it's some of these other more niche waste disposal systems that seem to be going wrong. Why is that important? Well, because there are already drugs that are in clinical trials to explore, you know, how we can reactivate these waste disposal systems to try get rid of this abnormal protein. And that means that once we know where the problem is as an individual, we can give personalized therapeutic approaches to target that specific problem. So, you know, if you think about, if the problem is that the waste isn't getting into the bin, well, fine, you develop a tool that's going to help get rubbish into the bin. But if the problem is that the bin's not being emptied, the last thing you want to do is give a medication that's just going to switch on waste disposal, because it'll just cause the overflowing of the bins. And so this is where we're at now. So we've just been really lucky to receive funding from the Alzheimer's Society to carry on the work that we've been doing, so that we can really pinpoint the problem now of whereabouts in these waste disposal systems the problem is. And also importantly, what we're trying to do now is identify something that a marker that we could measure in living patients with dementia, so that we can help people get into the right clinical trials. If we know someone has got a problem with their black bin, then they can go on a clinical trial, which is using drugs that help restore activity of that particular waste disposal system. So that's where the research is at now. So I'd like to just wrap up there and just thank my research teams for all the hard work that they've done over the years for making this work possible. And I'd like to obviously thank Alzheimer's Research UK and the Alzheimer's Society, because without that funding we wouldn't have been able to do any of the science that we've been doing. And obviously I need to thank Brains for Dementia Research and staff of the Manchester Brain Bank, but importantly all those people who are involved in the project, those who are enrolled, who have donated, and the families that have supported those decisions. So that's my stint over. So what I will do now is we're going to move to questions and answers, and I will stop sharing my slides.
Brilliant. Thanks so much, Gemma, for such an interesting talk. It's so great to hear about your work and the research that BDR is enabling to happen. And Fred as well, I was so sorry to hear about your wife, Joan. It's really sad to hear about what happened to her. And what you did for her was absolutely incredible, which I'm sure, yeah, we're all thinking about. So now we've heard from all of our speakers, we've got some time to answer your questions, so do keep them coming and we'll direct them at our speakers as appropriate. So starting off, we've got two questions from Rosemary Steele. So I think the first one is around how donors can have funerals when they die. Can donors have normal funerals? And how is it that the brain is harvested once a donor dies?
Yeah, I think I'll take that one, Leonie. So yeah, people can have a normal funeral after they've donated their brain. So the procedure is done in a way that the brain's taken out from behind the back of the head. So if people wanted to have a funeral and an open coffin, it's done in a way where you wouldn't be able to tell the procedure was done, which I know is a worry for some people, you know, if that's their wishes for the funeral. And how long we've got to get the brain donation done. We try and get it within 7 days, but really the sooner the better. Normally there's a bit of paperwork to do around registering the death, and at the moment, especially with COVID, you know, it can be quite difficult to get a slot at the registry office to actually register the death. So that can take a couple of days sometimes for us to receive the paperwork that's needed from registering the death. So I'd say on average, it probably takes about maybe 3 to 5 days to get everything arranged. But yeah, we try and fit it within those 7 days wherever we can. Okay,
great. Yeah, and on that note of COVID and the impact of COVID, have you been able to receive all of the brains from donors that have died during the period?
So at the start, as I said in my presentation, we sadly had to, you know, we were closed due to COVID at the beginning. you know, we had to figure out procedures for being able to get someone who could have potentially had COVID and you know, do the brain donation. And until we were able to get that process sorted and approved, you know, through the university, we were sadly not able to take donations until September. So you know, that was a real pity, really, because we did have families calling up, and you know, we weren't able to help them at that time. But yeah, as of September, it's been going really well. We rely on a local hospital to do the procedure of actually removing the brain. So sometimes it has been difficult to find a hospital at the mortuary to do the procedure, just because they've been so busy. But I think there's only been maybe one or two at the most that we've actually missed because, you know, we weren't able to find someone to do the procedure in time. Most of them we've been getting, and obviously now as things are calming down from the second wave, I think the mortuaries are being able to help us out more often. So
yeah, no, that's really good to hear that things are picking up again now, and hopefully that should continue over the next few months. We've just got another question from Rosemary about the impact of meditation. I'm just wondering if the work of the Manchester Brain Bank or Gemma, if you've been involved in any research relating to meditation, or if you know anything about it.
I haven't myself. I don't know about you, Gemma, if you've done any work around that.
No,
you're focused more on the pathology.
Yeah, I've not been involved in it myself, but there is a lot of evidence with respect to you know, the dementia risk, that any kind of behavioural initiative like meditation that is going to reduce stress and promote relaxation, reduce depression, is going to have a beneficial effect on the brain. We do a lot of outreach at the University of Salford, where we go to schools and we go to different community groups, and our main message is healthy heart, healthy brain. If whatever you can do to look after your heart and your brain, if you can, and you know that those things are hand in hand, then that is going to promote brain health, and that's going to reduce your dementia risk. So whilst I've not been involved myself, those are the kind of initiatives that we know are very good for brain health.
Okay.
Yeah, we do record. There's a lifestyle section as part of BDR where we record things like exercise, and usually that's where I record if, you know, people do meditation as part of their like weekly or monthly routine with regards to the lifestyle. And yeah, like Gemma said, you know, we know that stress, depression, lack of sleep all has an effect on cognition. So being able to do meditation, and if it helps those areas, you know, you could definitely see how that would help someone. And there has been research done around that's been looking into that effect.
Okay, great. So general health and including mental health is really important then.
Yeah.
Okay, I think we've got another question here for Gemma from Lynn Armstrong. She'd like to know if brain junk and the accumulation of proteins apply in multi-infarct dementia or vascular dementia.
Yeah, so you do get some buildup of proteins as well. And what we know, that there's a relationship between any vascular insult, any vascular disease, and the buildup of protein. So it's a bit of a vicious cycle. They kind of all relate to each other. The dementias aren't a clear-cut disorder. It isn't A causes B causes C. We know that there are numerous factors that work together to shape someone's risk for dementia. And this is why Brains for Dementia research is really important, because it allows you to consider all those things and how all those different factors work together. So there's variation in this junk. That's what I did my entire PhD on, was how this junk varies between different dementias and on an individual basis. Even if you get 10 people with Alzheimer's diagnosis, the amount of junk will be different, and the type of junk will be different, and the patterns might be slightly different. So there is individual variation, and that's because we're all different, and we've got these kind of various environmental and genetic factors that are all coming into play, and that's why the brain tissue is so useful, because it allows us to explore those really complicated interactions.
Okay, great. Yeah, that makes sense. And we've got another question about the buildup of brain junk as well from Kate Milne. She'd like to know if this happens in other neurological conditions such as MS.
It does, but in a different way. So in Alzheimer's, the junk accumulates in a much more predictable way. It affects different brain regions very sequentially, and that relates to how you see the disease manifest clinically. So the more junk you get in more brain regions, the more symptoms. Whereas in something like MS, you do get some accumulation of junk, but it's not as severe, and therefore it doesn't necessarily have it doesn't have the same effects. Whereas it's kind of, the junk is certainly driving the disease in dementia, whereas in other neurological disorders, it might be just occurring either as a byproduct of the various other bits and bobs that are going on in the brains of people with different disorders anyway. So.
Okay, great. We've got another question here from Kate asking about, so we've talked about the relationships between a healthy heart and a healthy brain and overall health and how that can help with preventing dementia. But is there any research on specifically gut bacteria and links to brain health or the buildup of brain junk?
Yes, there is. So this is quite a new area of research, and it's not my area of expertise at all, but it's very, very exciting how that there is increasing evidence that this kind of communication between a brain and gut, the brain gut axis, it's a thing, and there's a lot of people working on that now and seeing how gut health, and also it's not just gut health but oral health, and how our entire bodies, obviously our brain's main job is to coordinate everything, every system of our body, and so it's likely to have this kind of reciprocal relationship where the brain influences the body influences the brain, and we're certainly exploring all that. The field of dementia research really is so hopeful. There is so much work going on, and it's because Alzheimer's Research UK and Alzheimer's Society are funding amazing projects, and they are very variable as well. But yeah, it is going on.
Cool. Yeah, that's really exciting to hear about, definitely. Yeah, that's great. We've got another question here from Mary, which is about the links between aluminium and the possible consequences for developing brain disease. That was talked about quite a few years ago. She's wondering if there's been any updates on that in recent years.
I remember hearing about that, but I have to hold my hands up and say, yeah, I don't know what the result was. I know people were looking into quite a few different lifestyle factors. People who'd worked in industry where they might be exposed to things like that. But yeah, I wouldn't be able to say what the result was, I'm afraid. I don't know if anyone else has read any more into that.
Yeah, there is evidence that these number of metals, heavy metals, that are associated with dementia risk, and I guess it's it's a difficult area to really study and get to the bottom of, because you need to think about, well, how do you measure how much someone has been exposed reliably, and it's one of these things that it's quite easy for us to study in the lab. If we expose cells to stuff and then see if it kills them or not, we can do that, but it's quite difficult to understand the true meaningfulness of that it's very difficult to measure how someone's been exposed, how often, how the various other factors might be interplaying with that. So if you think about it, identifying causal relationships in dementia is really difficult, because you can say, right, okay, so there's evidence that aluminium causes some sort of damage and contributes to dementia. But how the process of being exposed could actually be something to do with a lifestyle change, and the exposure to the aluminium could be kind of coincidental, and it could be the lifestyle change underneath the aluminium exposure, and it's difficult to study. So there's more work needed, and I don't know how far that work has come along in its fruition.
Yeah, it's so complicated, and there's so many. It's hard to like separate all of the different factors, isn't it? Really, I guess. Yeah. Okay, thanks, Gemma. We've got another question here from Rosemary again. This is way above me. I do not understand what this means as a non-biologist. She would like to know if there's any correlation between the electronic frequency of healthy and deceased proteins in the brain.
I'm a biologist and I don't, that's above my head. That's a good question. I'll be honest, I'm not quite sure what is meant by that. We all have protein, we're all made of proteins and full of proteins, and we know that some proteins, they take on, especially when they're dying, they take on forms that aren't associated with our healthy self, and they can be damaging. And we also know that some genes have the ability, some gene mutations, for example, have the ability to switch off the good proteins, whereas some gene mutations switch on the bad proteins. And so it's about balance. We are quite, and our brains are quite adaptable. We can deal with a certain level of stress. And the issue seems to be when someone's normal equilibrium, when someone's normal balance gets tipped, so that the brain cells just can't cope with it anymore, and then they die. So a certain amount of stress, they say, don't they, a certain amount of stress is good for you, but then if you get too much, then you end up crying in a corner and anger eating custard creams, not speaking from experience or anything. But it's about balance, and the balance is driven by a huge number of really complicated systems, and that's the thing to bear in mind. And that's why brain tissue is really useful, because you can get a really good understanding of how these really complicated systems all interact with each other.
Okay, brilliant. Thanks, Gemma. So I've just looked at the clock and I think we're approaching 3 p.m. very shortly. So I'm going to wrap up the questions now and say a huge thank you to Phil and Fred and Gemma for your talks. It's so great to hear about all the work that's going on, and we're just understanding more and more about dementia each day. And it's so valuable to hear as well personal experiences that you've had, Fred, and hearing about your involvement with BDR. So thanks so much. A big thank you to everyone for attending this afternoon and giving your time, and for asking some really great questions there. I think we might have missed a couple of questions potentially. So the best thing to do to ask about these would be to get in contact with your local study team, who should be able to help. So the next of these events is confirmed for the 22nd of April, and this will be held by the BDR Centre. You won't need to sign up again to receive the joining instructions. These will be sent to you just a few days ahead of the event. So I'm going to close by thanking you on behalf of Brains for Dementia Research and for your ongoing involvement with the project. You're making such a valuable contribution to dementia research, and we really appreciate it. So I hope you're keeping safe and well and enjoying some spring sunshine, and looking forward to getting back to life as we used to know it. And I hope that you can join us again for the other events in the series. So thank you and goodbye.